Okay, let's unpack this. We're diving into a revolution that's happening in biology, specifically how we understand, the complex architecture of tissues.
Yeah.
If you're tracking the latest in cell biology, you've definitely heard of spatial transcriptomics, or ST.
Exactly. And if traditional sequencing tells us what ingredients are in a cell, it's gene expression. Spatial transcriptomics gives us that same data, but, and this is the critical part, it tells us where that cell is sitting within the tissue.
And that location, that context, is king, isn't it?
It absolutely is. I mean, biology is fundamentally spatial. Cells communicate based on who their neighbors are. And by defining these localized areas, we can identify what are called spatial domains. Think of them as specialized microenvironments or niches like the germinal centers in a lymph node or the regions right next to a tumor. To really understand disease, you have to know the address book.
Now, here's the problem, and it's a big one. As ST technologies like Xenium, Merscope, and Cosmax have gotten better and better, the data sets have become, well, a nightmare of complexity.
A total nightmare. We're talking millions of cells spread across dozens of slides, all run on different machines, maybe even using slightly different gene panels.
So that's the core challenge. How do you integrate what? Nearly 30 million cells across 18 completely different tissues, from the brain to the lung to the colon, and still pull out meaningful, unified knowledge.
And knowledge that isn't just an artifact of which machine ran the experiment. Existing tools, they just can't handle this scale and this diversity robustly. They just can't.
So our mission today is to introduce you to Novae. This is a graph-based foundation model, and it's designed specifically to solve this massive data integration headache.
And it was trained on that monumental 30 million cell data set to be a versatile zero-shot tool.
Its core promise, really, is eliminating the computational bottlenecks of older analysis methods by handling these experimental variations, these batch effects, natively and at incredible speed. To really appreciate what Novae is doing, I think we need to quickly look at the fractured landscape it's trying to unify.
Yeah.
You have sort of two main categories of ST tech.
You do. The first is next-generation sequencing-based, or NGS-based methods, things like Visium. They offer whole transcriptome data, so you get all the genes, but they're generally pretty low resolution.
Right, they're looking at spots, not necessarily single cells.
Exactly. You're analyzing expression at the spot level. Then you have the second category, the high-resolution, imaging-based techniques, Xenium, Merscope, all the big names.
And these give you that incredible subcellular detail.
Yeah, you can see exactly where molecules are, but the trade-off is they're limited to pre-selected gene panels. You might measure 500 genes instead of, say, 20,000.
So you get this mix. Low-res with all the genes versus high-res with just a few genes.
And that, plus the fact that researchers running 5 slides of breast cancer might use slightly different machines or tweaked panels, introduces that critical flaw we talked about, the batch effect.
Yeah, if my model finds a spatial domain, how do I know if it's a real biological niche or just a statistical echo of the fact that slide A was run on machine X and slide B was on machine Y?
Exactly. And the traditional methods are just so limited because they rely on these painful external two-step processes. First, they use a separate tool like Harmony for batch correction, a step that can take hours, sometimes days, on massive data sets.
Hours or days. Wow.
And then they pass that corrected data to another tool like Leiden or McClust to actually cluster the cells and assign the spatial domains.
So wait, if I want to adjust my resolution, let's say I want to look for 15 nuanced domains instead of 7 broad ones, I have to rerun those time-consuming external steps every single time.
Precisely. It's a huge analytical bottleneck.
Okay.
Novae was built to incorporate all of that complexity into one single massive learning model. It's what's called a self-supervised graph attention network, or GATE.
And gates are especially good at focusing on the most important neighbors in a network, right?
That's right, which makes them perfect for spatial data where all that local cell-to-cell signaling is just paramount.
So training Novae on 78 slides and 30 million cells means it's developed this kind of universal understanding of spatial biology. And that enables its key feature, zero-shot domain inference. You don't retrain it.
Nope, you just apply it to new data right out of the box. Now let's look under the hood at the core mechanism, because this is really where the speed and the flexibility are engineered. Novae starts by taking the cell expression data and converting it into a graph structure. It uses something called a Delaunay graph. Cells are the nodes and connections are the edges.
But is it looking at the entire tissue section at once? Because those can be huge.
No, and this is absolutely fundamental to Novae success. We immediately truncate any edges that are longer than 80 microns. Novae focuses intensely on the local microenvironment.
Okay.
It works by sampling small subgraphs, usually between 15 to 80 cells, based only on those nearby connections.
So you're intentionally zooming in on the immediate neighborhood. Why is that local focus so important?
It prevents something called oversmoothing. If you aggregate information over too large an area, all the detailed biological signals just blur together.
Right, like taking a photo with motion blur.
Exactly. By focusing locally, the model keeps that fine-grained resolution you need to define those tiny microniches.
That makes perfect sense for spatial context. But let's go back to that panel problem. So I'm a researcher. I only ran 500 genes on my slide. But the model was trained on data with 5,000 genes.
Yeah.
If we don't share genes, how can it possibly compare my cell data to its huge training database?
This is where the panel invariant embedding is, well, it's revolutionary. Instead of looking for shared gene lists, which is the old way, Novae translates the gene expression into a consistent vector space using trainable gene embeddings.
Wait, like a biological translator?
Exactly that. Think of it this way. Novae isn't reading the individual words, the genes. It's identifying the underlying biological programs or concepts that they represent. It keeps it all consistent using L2 normalization, which just ensures the weights are comparable even if one slide has 10 times more measured genes than another.
That is huge.
Yeah.
It completely removes that intersection of genes problem that has plagued cross-study analysis for years.
It does.
And you make the model even tougher during training by adding noise.
We do. We apply two specific augmentations. First, we introduce pseudo-batch effect noise. It's basically simulated experimental jitter, which teaches the model to ignore real-world variations from different machines. Second, we use random gene panel subsetting, where we just randomly drop a ratio of genes.
Ah, so you're simulating running studies with different limited panels.
Exactly, which further reinforces that panel invariant learning.
Okay, so we have these localized, robust, and translated subgraphs. How does this graph attention network actually learn the identity of those spatial domains?
This is where we fuse the self-OAV framework with optimal transport. SESSO-AV is a self-supervised method designed to learn structure by agreement. It looks at two slightly different views of the same local neighborhood, our subgraphs, $0 and $0, and it expects their representations to belong to the same underlying domain.
Okay.
To do that, Novae learns prototypes. You can think of these as high-resolution learned embeddings that represent the fundamental building blocks of all spatial domains.
So the prototypes are the foundational knowledge bricks, the dictionary of tissue microenvironment.
Precisely. Now, here is the secret sauce for the speed in the batch correction. Optimal transport, or OT.
Okay.
Imagine OT as a dynamic traffic regulator for the data flowing into the prototypes. If a mini batch of cells from, say, slide A is always being assigned to prototype 1 and it's ignoring prototype 2, then that slide is introducing a bias. It's a mode collapse.
So it's hogging the prototype assignment.
It's hogging it, yes. Optimal transport steps in and uses the assignments to calculate a corrected assignment. This calculation ensures that every mini batch uses all the prototypes evenly. It's dynamically coordinating the data flow.
And by doing that,
OT eliminates the batch effect right there in the training loop, completely replacing the need for those slow external tools like Harmony.
wow. And the final piece is the agreement part.
That's the swapped cross-entropy loss. It takes the corrected assignment from one subgraph and forces the probability from the other subgraph to match it, and then it swaps their roles. This forces the two views of the same neighborhood to agree on their identity, ensuring the model is learning real biological structure and not just noise.
That was dense, but incredibly rewarding. Let's just hit the pause button for a second and distill the technical genius we just covered into 3 takeaways for you, the learner.
Good idea.
First, spatial context is king, but local focus is queen. Novae uses graph attention on local subgraphs to understand those microenvironments, and that avoids the blur of oversmoothing. Second, panel invariant learning is the bridge. By using these trainable embeddings, Novae can compare 2 slides no matter how many genes they measure. It finally solves that cross-study problem.
A huge one.
And third, self-correction is speed. Optimal transport and prototypes natively handle the batch correction, which eliminates those days-long bottlenecks of external tools.
A perfect summation.
Which brings us to the flexibility problem. We learned Novae created these tiny foundational prototypes. But if I'm a pathologist looking at a whole tissue section, I'm interested in the big, coarse-grained structures. So how does Novae translate those tiny prototypes into the major domains we actually study?
That's the beautiful design choice that really unlocks Novae's speed. The prototypes are treated as the leaves of a hierarchical tree. So when a user requests a specific number of domains, say 7 overall tissue domains, Novae simply applies hierarchical clustering to the prototypes themselves.
So changing the resolution of your analysis, going from, say, 7 domains to 15 highly specific domains, is instantaneous.
Essentially, yes, it's constant time. Since the prototypes are already learned and organized, switching resolutions is just a matter of moving up or down this pre-calculated tree.
Right, whereas competitors have to rerun those slow clustering algorithms like Leiden every single time.
Every time. It's a huge difference.
That brings us back to the core advantage here, efficiency. We saw that for some competitors, that batch correction and clustering step on a big data set like 6 million cells can take up to days. Novae just eliminates that waiting period.
We're talking about processing time shrinking from days to mere seconds, all because the model handles those complex operations internally. And crucially, efficiency includes memory management. By using lazy loading and only focusing on local subgraphs, Novae keeps VRAM and RAM usage low. This allows researchers to actually work with these monumental 30 million solid datasets without needing, a supercomputer.
Now, let's talk robustness. If it's integrating 18 different tissues, how do you prevent the model from finding commonalities that aren't actually real? You don't want a breast tumor domain showing up in a healthy piece of colon tissue.
Right. You have to balance the integration with biological realism, preventing what we call overcorrection. The optimal transport constraint is actually slightly relaxed using a queue system. This makes sure that while Novae finds generalized domains shared across many tissues, like basic immune niches, it also allows tissue-specific prototypes to remain unused on slides where they just don't belong.
So it maintains the distinct biological identity of each sample. How did the testing prove that?
Well, in a mouse brain robustness case study, even when a large region of tissue was artificially removed from one slide, Novae maintained high domain continuity. We measure this with something called the FIDE score. A high FID score means the learned domain boundary stays smooth even with missing data.
Nice.
It also kept a low post-split JSD score, which confirms it didn't artificially blend unrelated tissue types together, so it resisted that overcorrection perfectly.
And it's robust against the segmentation method, too, since it worked regardless of whether the input was from the 10x genomics default or the Bayes method. That's some serious stability. Let's move to the payoff, the biological insights this new engine revealed. Let's start with the human lymph node architecture. What kind of structural discoveries did Novae enable when comparing a non-diseased node to a reactive one?
This is a perfect example of how that spatial context changes everything. Using spatial trajectory analysis on the Novae representations, researchers saw this dramatic structural reorganization in the reactive node.
Okay.
Specifically, the germinal center domain, that's the hub for mature B cells, it became highly connected to 5 other domains compared to only 1 connection in the healthy slide.
Wow, that sounds like a complete architectural shift in response to inflammation.
It is. And they also noted the expansion of a new domain, D498, which was highly enriched in a chemoattractant called CXCL12. This strongly suggests that the model isolated and highlighted a newly formed inflammatory cell recruitment area, which is crucial to the node's immune response, a dynamic change that older, batch effect-prone tools likely would have just missed.
Shifting to cancer, Novae also showed its flexibility by analyzing spatial protein data from Cosmx in head and neck cancer. What did it find regarding that favorable prognosis biomarker, the multinucleated giant cells, or MGCs?
Novae found that MGC-positive patients, so those with a better clinical outcome, had a significant reduction in a specific stromal-like domain, D497.
And why does that matter?
Because D497 was enriched in regulatory key cells, or TREGs. Since TREG cells are known to suppress the immune system, the model is essentially showing us that patients with a good biomarker, the MGCs, are physically losing their local immunosuppressive environment. It links spatial organization directly to clinical prognosis.
So that ability to connect location, cell type, and outcome, that's the ultimate goal of ST. So finally, let's talk about the future. Multimodality. Novae can integrate H&E morphological data with transcriptomics. How does it technically achieve that fusion?
It's actually really elegant. It brings in a separate pathology foundation model, something like CONCH, to compute visual embeddings from the H&E image patches. Novae then fuses these visual embeddings with its own transcriptomic graph representations.
And the result?
The combined model achieved the highest FIDE scores, indicating enhanced domain specificity.
Can you give an example of the kind of separation you only get with that added morphological context?
Yeah, in lung tissue, the multimodal approach was able to cleanly separate two domains, D2032 and D2027, that Novae alone had kind of merged. This separation was driven entirely by the distinct morphology of the bronchus, that small airway structure, which was captured by the H&E image.
So the visual data gave the model the necessary anatomical detail to resolve a separation the gene expression alone couldn't reliably confirm. So Novae provides this universal, fast spatial foundation that supports pretty much every downstream task, from finding spatially variable genes like Nero6 in the aging mouse brain, to pathway analysis, to structural discovery.
It's the analytical hub that spatial biology has really been waiting for. It moves ST analysis beyond these single-slide, fixed-panel studies into a generalizable pan-tissue foundation model, leveraging the power of self-supervised learning, optimal transport, and that hierarchical prototype architecture.
While Novae is architecturally ready for anything, you did mention one current limitation regarding its pre-trained models.
That's right. Currently, the pre-trained Novae models rely exclusively on transcriptomics. Now, this isn't a limitation of the software itself, but of the data landscape. We just lack these large-scale, combined, multimodal data sets that include transcriptomics, proteomics, and histopathology, all on the same slides across a massive range of tissues.
But once that data becomes available,
The model is designed to absorb it.
this foundation model approach is incredibly powerful for biological discovery, especially when you're dealing with degraded or partial data. We saw Novae was robust, but its performance did start to drop when about 60% of the tissue or gene expression was lost. Considering that the model can pinpoint disease pathways and structural changes using these foundational domains, here's my final thought for you, the learner, to mull over. If reliable clinical insight depends on the model ignoring experimental noise but maintaining biological signal, what level of tissue degradation or information loss do you think represents the boundary between gaining reliable, clinically relevant insight and just reading noise?